Welcome back, everyone, to The Deep Dive. Today, we're really jumping into the deep end with the fourth basic tissue type.
That's right. We've covered epithelia, connective tissue, nervous tissue.
Exactly. And now it's time for the engine room, muscle tissue.
The powerhouse.
Our mission today, really, is total mastery. We want to get into the histology, the details that, separate just knowing about muscle from truly understanding it, like the pros.
Couldn't agree more. Because fundamentally, if you think about movement, pumping blood, even just standing upright,
Yeah.
it all comes back to muscle.
And that single property.
Contractility. Precisely. Contractility. It's the universal cellular superpower here, optimized in every single muscle cell.
So when we say contractility, we're talking about generating force at the molecular level.
Exactly. And the machinery is surprisingly consistent across the board. It relies on these protein partners, actin microfilaments.
And the myosin proteins associated with them, little motors.
Tiny, highly specialized motors, yeah, driving everything from lifting a weight to your heartbeat to squeezing food through your gut.
It's amazing. But even with that core mechanism, nature's given us three distinct flavors, right? Each adapted for its job.
Yep, our three pillars, based on structure, function, how they look under the scope.
Okay, let's lay them out. Pillar one, skeletal muscle. This is the one most people picture, right? The biceps, the quads.
The striated titan, as you call it, yeah. Long cells, cylinders, really. Multinucleated, and those very obvious cross striations.
And key features. Quick, powerful contraction, and usually under our conscious voluntary control.
Usual, yes. Then pillar two, cardiac muscle, the heart.
Still striated, so it looks a bit similar in that way.
It does, shares the striations.
Yeah.
But the cells are different. Elongated, often branched, kind of forming a network.
And the big giveaway visually.
The intercalated discs, those unique junctions between the cells, you see those, you're looking at heart muscle.
Functionally. Gotta be involuntary. Vigorous rhythmic pumping, nonstop.
Absolutely. And finally, pillar three, smooth muscle,
Yeah.
the quiet workhorse.
Right. Found in the walls of organs, blood vessels.
Exactly. These cells are different again. Fusiform, kind of spindle-shaped, tapered ends, and the big visual clue. No striations at all.
Hence the name, smooth.
Hence the name. Their job is slow, steady, continuous contraction. Totally involuntary. Think digestion, blood pressure regulation.
Okay, three distinct types. Before we dissect them further, we need the lingo, the specialized vocabulary.
Ah, yes, the sarcos terms. Essential if you want to navigate muscle histology properly. Comes from the Greek for flesh, sarcos.
So instead of just saying cytoplasm in a muscle cell.
You say sarcoplasm. Emphasizes its unique, packed with protein nature.
Got it. And the smooth ER, which we know is vital for calcium.
Becomes the sarcoplasmic reticulum, or just SR for short. It's hyper-specialized for calcium handling in muscle.
And the cell membrane plus its outer coating.
That whole package is the sarcolemma. Using these terms just instantly signals, the specifics of muscle cells.
Good to know. And developmentally, where do they all come from?
Overwhelmingly, mesoderm. Almost all muscle cells trace back to that middle embryonic layer.
And development involves what? Cell lengthening? Making proteins?
Both. Huge amounts of lengthening and just monumental synthesis of actin and myosin.
Yeah.
That's what fills up the sarcoplasm.
Okay, so they develop. How do muscles grow bigger, like after we work out? There are two main ways, right? Hypertrophy and hyperplasia.
Exactly. And it's crucial to distinguish them. Hypertrophy is the big one for skeletal muscle.
That's when the individual cells get bigger.
Precisely. Increase in cell volume. Exercise stimulates the cell to make more myofibrils, those internal contractile bundles, so the existing fiber swells in diameter. No new cells.
Just bigger, beefier cells.
Right. But hyperplasia is different. That's an increase in the actual number of cells through mitosis, cell division.
And skeletal muscle is bad at that.
Terrible. Cardiac muscle, too. They're too complex, too integrated. But smooth muscle.
They can do it.
Oh, yeah. Smooth muscle cells are simpler, single nucleus. They readily divide. That's how organs like the uterus can grow so rapidly during pregnancy. Lots of hyperplasia. It's a key versatility.
Okay, let's really zoom in on skeletal muscle now. The striated titan. You said these fibers are giants.
Relatively speaking, yes. Long cylinders, multinucleated. Diameters can range quite a bit, maybe 10 micrometers, up to a hefty 100 micrometers.
And that multinucleated thing comes from their development.
Exactly. It starts with these precursor cells, myoblasts from the mesenchyme. They line up, fuse end to end.
Like building a long chain.
Precisely. They fuse to form these myotubes, which are essentially immature multinucleated muscle fibers. This fusion explains the length, obviously.
And also why there are so many nuclei and where they end up.
You got it. As the myotube matures and fills with contractile proteins, all those nuclei get squeezed out to the very edge, right under the sarcolemma.
So finding peripheral subsuccolemmal nuclei is a dead giveaway for skeletal muscle under the microscope.
It's one of the classic tells. Definitely.
Now, these fibers are complex. Can they repair themselves if damaged? Is there any regeneration?
There is, but it's limited. And it depends entirely on a small, quiet population of cells called muscle satellite cells.
Satellite cells, like orbiting the main fiber.
Kind of. They're basically inactive myoblasts, reserve stem cells, tucked away right next to the muscle fiber under its external lamina.
So if there's minor damage.
These satellite cells can wake up, start dividing, and fuse to either patch the damaged fiber or even form short new segments. It's crucial for minor repairs.
But major injuries.
Overwhelm this system, you get scar tissue formation instead, so limited regeneration potential.
Okay, now organization. A single fiber contracting is one thing, but a whole muscle needs structure to work. You mentioned connective tissue layers.
Absolutely critical. It's not just random packing. There's a precise hierarchy of connective tissue wrappings, very much like you see in a peripheral nerve, actually.
Okay, starting from the outside of the whole muscle.
That's the epimysium. Epi means upon. It's a dense, irregular connective tissue sheath that covers the entire muscle. Think of it as the tough outer jacket.
And it carries the main supply lines in, nerves, blood vessels.
Exactly. Larger vessels and nerves travel within or penetrate through the epimysium to get inside.
Then what? Does it send layers inward?
It does. These partitions, or septa, extend inwards and surround bundles of muscle fibers. This layer is the perimysium. Peri means around.
And the bundle it surrounds, it's called?
A fascicle. This is a really important functional unit. Often, a single nerve branch will supply a fascicle, so all the fibers within it tend to work together.
So the perimysium groups fibers into these functional teams. Makes sense for coordinating movement.
Precisely. Allows for graded control. You can activate different fascicles within a large muscle.
Okay, epi-peri. What's the innermost layer right around the individual fiber?
That's the endomysium. Endo means within. This is the most delicate layer. Just a thin mesh of reticular fibers, a few fibroblasts surrounding the external lamina of each single muscle fiber.
And this is where the immediate support system is. Capillaries, nerves.
Exactly. The endomysium is rich with capillaries delivering oxygen and nutrients right to the fiber's doorstep. And the very fine terminal nerve branches travel through it too.
So epimysium around the whole muscle, perimysium around fascicles, endomysium around individual fibers.
That's the hierarchy. And importantly, all these layers are continuous with each other. They're not separate things. They merge.
Why is that continuity important?
Force transmission. The contraction starts deep inside the fiber, but that force needs to get out to pull on something, usually a bone. These connective tissue layers collect and channel that force.
Leading to the connection point, the tendon.
Exactly. The myotendinous junctions. Myo for muscle, tendinous for tendon. This is where all those connective tissue layers, epi, peri, endo, plus the deep fascia around the muscle blends seamlessly into the dense, regular connective tissue of the tendon.
How does that connection work at the microscopic level? Is it just glued together?
It's much more intricate. The collagen fibers from the tendon actually interdigitate. They insert themselves amongst the muscle fibers. And the sarcolemma at the very end of the muscle fiber is thrown into deep folds.
Folds to increase surface area.
Massively increased surface area. The collagen fibers associate with these folds. It creates an incredibly strong bond, distributing the pulling force, so the muscle doesn't just tear away from the tendon during powerful contractions. It's a really robust mechanical linkage.
Okay, we've got the fiber organized. Now let's dive inside the fiber into the sarcoplasm. You said it's packed with these bundles, myofibrils.
Yeah, thousands of them, potentially. Long cylindrical bundles running parallel, filling up most of the cell volume.
And the striations we see, the stripes, they come from these myofibrils.
Directly. It's the highly ordered, repeating arrangement of proteins within each myofibril, and the fact that all the myofibrils are aligned in register, side by side. That creates the banded appearance.
Okay, the bands. There are dark ones and light ones.
Right. The dark bands are called A bands. A for anisotropic, meaning they mess with polarized light because they're dense and highly ordered.
And the light bands?
Are the I bands. For isotropic, they don't significantly alter polarized light. They appear lighter.
And this pattern repeats?
Over and over. The fundamental repeating unit, the structural and functional unit of contraction, is the sarcomere.
Defined as?
Stretching from one Z disk to the next Z disk. The Z disk, or Z line, is that dark line you see running right down the middle of each light I band.
How long is one sarcomere? roughly?
In resting muscle, about 2.5 micrometers.
Yeah.
Tiny, but billions of them contracting in series and in parallel generate incredible force.
Okay, 2.5 micrometers of highly organized machinery. Let's break down what's inside. The main players are the thick and thin filaments.
Absolute stars of the show.
Yeah.
Thick myofilaments and thin myofilaments.
Let's start thick, made of.
Primarily the protein myosin. These filaments are about 1.6 micrometers long, 15 nanometers thick. They occupy the A band, the dark band.
And the myosin molecule itself is complex.
Huge. Around 500 kilodaltons. It's got two heavy chains twisted into a long tail.
And then heads sticking out.
Exactly. Globular heads project off the tail at one end. Each head has associated light chains too, and these heads are the business end.
Because they have binding sites.
Two crucial ones per head. One site binds to actin on the thin filaments as forms the cross bridge. The other site binds ATP, the energy currency.
And it uses that ATP.
Yes, the head acts as an enzyme, an ATPase, specifically actin-myosin ATPase activity. It hydrolyzes ATP to release energy, which powers the contraction stroke.
How are these arranged in the thick filament? Random.
Oh no, highly organized. Hundreds of myosin molecules bundled together with their tails pointing towards the center of the sarcomere and their heads sticking outwards towards the ends, ready to grab onto actin.
Okay, now the thin myofilaments. Thinner, shorter.
Yep, about 1 micrometer long, only 8 nanometers wide. Made primarily of the protein actin, specifically F-actin, which is a polymer of G-actin monomers.
F for filamentous, G for globular.
You got it. The thin filament is basically two strands of these G-actin beads twisted into a helix.
And each G-actin bead, does it have a binding site?
It does. Each G-actin monomer has one specific site where a myosin head can bind. But
There's a catch.
there's a regulatory system controlling access to that site. It's not always available.
Ah, the guards. What are they?
The troponin-tropomyosin complex. First, you have tropomyosin. It's a long, thin, coil-like protein molecule, about 40 nanometers. It lies right in the groove formed by the twisted actin strands.
And its job is?
In a resting muscle, it physically covers up the myosin binding sites on the actin molecules. It blocks the interaction.
Okay, so tropomyosin is the blocker. What about troponin?
Troponin is a complex of 3 smaller proteins attached to the tropomyosin molecule at regular intervals. These 3 subunits have very specific jobs. You really need to know these for mastery.
Yay them out for us.
Okay, there's TNTT for tropomyosin. This subunit binds the whole troponin complex to tropomyosin. Then there's TNII for inhibitory. This one helps hold tropomyosin in the blocking position, inhibiting the actin-myosin interaction.
And the third one, the really critical one.
That's TNCC for calcium. This subunit has binding sites for calcium ions when calcium levels rise in the sarcoplasm.
It binds to TNC.
Exactly. And that binding event triggers a shape change in the whole troponin complex.
Which moves the tropomyosin.
Precisely. It pulls tropomyosin off the actin binding sites, exposing them so myosin heads can attach. Calcium binding to TNC is the molecular switch that turns contraction on.
Incredible detail. Okay, let's map these filaments onto the bands we see. The light I band.
Contains only the thin actin filaments. They extend from the Z-disc inwards.
And they're anchored at the Z-discs.
Yes, anchored perpendicularly by a protein called onactinin, which forms the structural backbone of the Z-disc.
Are there other important proteins in the I-band Z-disc area?
Absolutely. 2 giant ones are crucial for structure and elasticity. First, titin.
The biggest protein in the body, you mentioned?
Literally, around 3,700 kilodaltons. It's enormous. Titin filaments run from the Z-disc all the way to the thick filaments.
What does it do? Act like a spring?
Exactly like a molecular spring. It provides elasticity, helps the sarcomere recoil after stretching, keeps the thick filaments centered within the sarcomere, and prevents overstretching. Hugely important for passive tension.
Wow. And the second one?
Nebulin, another large protein, but this one runs alongside the thin filaments. It's thought to act like a molecular ruler.
A ruler?
Yeah, helping to specify the precise length of the thin actin filaments as they're assembled during development. Ensures uniformity.
Okay, so I-band is thin filaments plus titin and nebulin anchored at the Z-disc. What about the dark A-band?
The A-band contains the entire length of the thick myosin filaments. Plus, in the outer parts of the A-band, it includes the regions where the thin filaments overlap with the thick filaments.
It makes sense why it's darker, more protein density there. Is there a region in the middle of the A-band that's slightly lighter?
Yes, when the muscle is relaxed, there's the H zone. H for heller, meaning brighter in German. This central part of the A-band contains only the thick filaments specifically, just the bare rod-like tails of the myosin molecules where there are no heads and no overlap with thin filaments.
And right down the middle of the 8 zone.
Is the M line.
Mhm.
M for middle, middle. This line contains proteins like myosin that link the thick filaments together, holding them in their precise hexagonal lattice arrangement.
Any functional role beyond structure for the M line?
Yes, it's also packed with the enzyme creatine kinase.
Ah, for energy.
Right. Creatine kinase takes phosphate from phosphocreatine, a high-energy storage molecule in muscle, and quickly adds it to ADP to regenerate ATP right there, close to the myosin heads that need it urgently at the start of contraction.
It's amazing. Even with giants like Titan and Nebulin, myosin and actin are still over half the protein in muscle.
Easily. The sheer density of the contractile machinery is phenomenal. It's built for power.
Okay, powerful machinery needs a rapid, synchronized start signal. How does the nerve impulse at the surface trigger all these internal myofibrils at once?
Through the triad system. It's a brilliant piece of cellular engineering.
Involving the sarcoplasmic reticulum.
Definitely. The SR is this intricate network of membrane tubules surrounding each myofibril like a lace stocking. Its main job is to pump in and store huge amounts of calcium ions and then release them on command through voltage-gated C2+ channels.
But the command starts at the surface, the sarcolemma. How does it reach the SR deep inside?
Via the transverse tubules or key tubules. These are deep finger-like invaginations of the sarcolemma itself. They dive down from the surface and wrap around each myofibril.
Where exactly do they run?
Critically, they run right near the boundary between the A-band and the I-band. And this positioning is key.
Because that's where they meet the SR.
Precisely. Each T-tubule comes into close contact with two enlarged sacs of the SR, called terminal cisternae.
One T-tubule plus 2 terminal cisternae.
That specific structural arrangement is called the triad. It's the physical link. The depolarization wave traveling down the T-tubule membrane directly triggers the opening of the calcium channels in the adjacent SR cisternae.
So the signal jumps from T-tubule to SR, releasing calcium everywhere almost instantly.
Simultaneously throughout the entire muscle fibrous cross-section. That's how you get a rapid, coordinated, forceful contraction of all myofibrils together. The triad is the key to excitation-contraction coupling in skeletal muscle.
All right, we have the structure, the proteins, the signal system. Let's walk through the actual movement, the contraction cycle. The core idea is the sliding filament model.
Yeah, absolutely fundamental. You have to grasp this. The filaments themselves, actin and myosin, do not shorten. They slide past each other.
So the overlap between them increases.
Exactly. The thin filaments are pulled inwards towards the center of the sarcomere over the thick filaments. This pulls the Z discs closer together, shortening the entire sarcomere and thus the myofibril and the muscle fiber.
Okay, step by step. What's the resting state?
In rest, calcium levels in the sarcoplasm are very low because the SR has pumped it all away. And importantly, the tropomyosin molecules are blocking the myosin binding sites on the actin filaments. Myosin wants to bind, but it can't.
Then comes a signal.
Nerve impulse arrives, travels down the T-tubules, hits the triad.
And triggers massive calcium release from the SR.
Yep. Calcium floods the sarcoplasm.
And this is the trigger for activation.
Right. Calcium ions find and bind to troponin C, TNC, on the thin filaments.
Causing that shape change we talked about.
Exactly. The whole troponin complex shifts, pulling tropomyosin with it out of the groove. Suddenly, those myosin binding sites on actin are exposed, open for business.
Now the crossbridge cycle can begin.
Yes. The energized myosin heads, they've already split ATP from the previous cycle, immediately lock onto the exposed actin sites, forming the crossbridge.
Step 1, attachment. Then what?
Step 2, the power stroke. Binding to actin triggers the myosin head to pivot or swivel forcefully. As it pivots, it pulls the attached thin filament inwards towards the M line. This is the actual force-generating step. ADP and phosphate are released during this stroke.
Pulling the Z discs closer. Okay, now the head is bound and pivoted. How does it let go to repeat the cycle?
Step 3, detachment. A new molecule of ATP has bind to the myosin head. This binding causes the head to detach from the actin filament.
So ATP binding causes detachment. Interesting.
Crucial point. Then step 4, re-energizing. The ATP bound to the myosin head is immediately hydrolyzed back to ADP and inorganic phosphate, pi. The energy released by this hydrolysis cocks the myosin head back into its high-energy pre-pivot position, ready to bind to another actin site further down the filament, provided calcium is still present and the site is exposed.
And the cycle attach, pivot, detach, re-energize repeats.
Over and over, as long as calcium and ATP are available. Each cycle inches the thin filament along a tiny bit. Hundreds of these cycles happening rapidly in all the sarcomeres generates the macroscopic shortening.
How fast is one cycle?
Very fast, maybe around 50 milliseconds or so,
Yeah.
but it happens thousands, millions of times across the fiber.
Okay, how does it stop? Relaxation.
The nerve signal stops. The SR actively pumps calcium back inside using ATP-powered pumps. Calcium levels in the sarcoplasm drop rapidly.
Calcium detaches from TNC.
Yes, without calcium bound, troponin reverts to its original shape, pushing tropomyosin back into the blocking position on the actin filaments.
Myosin can't bind anymore.
Can't form cross bridges. The contraction stops, and the muscle fiber passively lengthens back to its resting state, partly due to the elastic recoil from proteins like titin and external forces.
This cycle perfectly explains rigor mortis, doesn't it?
It really does. After death, cell metabolism stops. No more ATP is produced.
Just so myosin heads can attach and pivot.
But without new ATP binding, they can't detach from the actin. Step 3 fails.
They get stuck in the attached state.
Exactly. All the myosin heads become locked onto actin, forming stable, rigid cross bridges. That's what causes the stiffness of the muscles. It's a direct consequence of the lack of ATP needed for detachment.
Fascinating and a bit grim. Okay, let's back up to the signal source, the neuromuscular junction, NMJ, the synapse between nerve and muscle.
Right, the NMJ, or sometimes called the motor end plate, MEP. It's a highly specialized chemical synapse.
So a motor nerve fiber approaches the muscle fiber.
A myelinated axon branches near its target fibers. The very end loses its myelin sheath and forms these little swellings, the axon terminals.
And these terminals sit in little depressions on the muscle surface.
Yes, in specialized troughs on the sarcolemma. The axon terminal is packed with synaptic vesicles,
Containing?
the neurotransmitter acetylcholine, or HE.
Okay, nerve impulse arrives at the terminal.
Triggers the vesicles to fuse with the axon membrane, releasing HE into the narrow space between the nerve and muscle, the synaptic cleft.
What happens on the muscle side, the postsynaptic side?
The sarcolemma here is dramatically folded into deep junctional folds.
More surface area again.
Exactly. Maximizes the area packed with acetylcholine receptors. ACE diffuses across the cleft and binds to these receptors.
What do the receptors do when acetylcholine binds?
They are ligand-gated ion channels. Binding opens them, allowing sodium ions, Na+, to rush into the muscle cell and potassium ions, K+, to trickle out. The net effect is a rapid influx of positive charge.
Depolarization.
Depolarization of the sarcolemma at the end plate. This creates the muscle action potential, which then propagates along the sarcolemma and down the T-tubules.
How is the signal turned off? We need muscles to relax quickly.
Crucial point. The ACE in the synaptic cleft is very rapidly broken down by an enzyme called acetylcholinesterase. It floats in the cleft and chews up ACE almost as soon as it's released.
Preventing continuous stimulation.
Exactly. Ensures the signal is brief and precisely controlled, allowing for rapid cycles of contraction and relaxation.
Now, how does the nervous system control the force of contraction? It's not always maximum effort.
Through the concept of the motor unit.
Define that again.
A motor unit is one single motor neuron, the nerve cell in the spinal cord, and all the individual muscle fibers that its axon branches innervate. Could be just a few fibers, could be hundreds.
And the key principle here is?
The all or none principle for the muscle fiber itself. When a motor neuron fires, all the muscle fibers in its motor unit contract maximally. They don't do partial contractions.
So if a fiber contracts, it contracts fully. How do we get graded contraction then? Different levels of force.
By controlling two things. First, the number of motor units activated, recruitment, and second, the size of the motor units being activated.
So activating more motor units means more fibers contracting, thus more force.
Precisely. And activating larger motor units, those innervating hundreds of fibers, generates more force than activating small motor units, innervating just a few fibers.
Which explains fine control versus power, like eye muscles versus leg muscles.
Perfect example. Eye muscles need incredible precision. Their motor units are tiny, maybe one nerve controlling 5-10 fibers. Your big leg muscles need power. Their motor units are huge, one nerve controlling maybe 1,000 or more fibers. Recruitment of these different units allows the smooth, graded force you need for any action.
This NMJ system is so precise. What happens if it goes wrong? You mentioned myasthenia gravis.
Yes, a classic autoimmune disease targeting the NMJ. The patient's own immune system produces antibodies that attack the acetylcholine receptors on the muscle side.
So the receptors get blocked or destroyed.
Both. The antibodies interfere with AC binding, and they also trigger the muscle cell to internalize and degrade the receptors. The number of functional receptors plummets.
Leading to?
Failed neuromuscular transmission. The muscle doesn't respond properly to nerve signals. This causes characteristic fluctuating, activity-dependent muscle weakness. It often starts in the eye muscles, causing droopy eyelids, petiboctosis, and double vision diplopia because those muscles are working constantly.
Debilitating. Okay, one last thing on skeletal muscle fiber types. They aren't all the same physiologically.
Not at all. They're specialized for different tasks, primarily based on how fast they contract and how they make ATP. And interestingly, the type of nerve that innervates a fiber determines its type. So all fibers in one motor unit are the same type.
What are the main types? I remember red and white muscle.
That's the classic distinction relating to myoglobin content. We usually classify them into three main categories now based on speed and metabolism.
Okay, type 1.
Slow oxidative fibers, type I. These are the red fibers. High myoglobin stores oxygen, packed with mitochondria, dense capillary network.
Built for endurance.
Exactly. They contract relatively slowly, but rely on oxidative phosphorylation, aerobic metabolism, for ATP, making them very resistant to fatigue. Think postural muscles, marathon running muscles.
Okay, type 2.
There are subtypes here, but the extreme is fast glycolytic fibers, type I, or sometimes IX in humans. These are the white fibers. Low myoglobin, fewer mitochondria, fewer capillaries, but lots of stored glycogen.
Built for speed and power.
Yes, they contract very rapidly, high myosin ATPase activity. They rely heavily on anaerobic glycolysis for quick bursts of ATP.
But they fatigue quickly.
Very quickly, because glycolysis produces lactic acid, which builds up. Think sprinters, weightlifters, eye muscles making quick movements.
Is there an intermediate type?
Yes, the fast oxidative glycolytic fibers, type 2I. They have properties of both. Reasonably fast contraction, but also good oxidative capacity, making them more fatigue-resistant than IOP fibers. They're adaptable, used in activities needing both speed and some endurance.
So slow endurance, fast power, and an intermediate. The mix varies between muscles.
Absolutely. Your postural back muscles are predominantly type 1. Muscles for sprinting or jumping are rich in type IBIX. Most muscles are a mix, and training can shift the proportions somewhat, especially between the type 2 subtypes. Understanding these fiber types is key to exercise physiology and training.
Let's just now to the heart. Cardiac muscle. You said it's striated like skeletal, but the cells are different.
Right. Still has the sarcomeres, the A and I bands, but the cells themselves, the cardiomyocytes, are shorter, typically 15-30 micrometers wide and 85-120 long, and they often branch.
Branching, so they connect to multiple neighbors.
Exactly. They align end-to-end, but also side-to-side via branches, forming these intricate, interwoven, spiraling bundles. This structure is perfect for the squeezing, twisting contraction the ventricles perform to eject blood.
And the nucleus. Skeletal had many peripheral ones.
Cardiac cells usually have just one central nucleus, sometimes two, but typically one right in the middle of the cell. Another key difference.
But the absolute defining feature you said is
The intercalated discs. These are unique to cardiac muscle. You see them as dark, somewhat irregular lines running transversely across the fibers where two cells meet end to end.
What are they actually made of? Just glue?
Much more complex. They are specialized intercellular junctional complexes. Think of them as having two main components based on their orientation. The parts of the disc that run transversely, perpendicular to the pulling force, are packed with anchoring junctions, desmosomes and fascia adherens junctions.
For strength.
Pure mechanical strength. Desmosomes link intermediate filaments between cells. Fascia adherens link the actin filaments of the terminal sarcomeres to the membrane. They basically rivet the cells together, preventing them from pulling apart during the heart's constant, powerful contractions.
Okay, strong mechanical links. What about the other parts of the disc?
The parts that run longitudinally, parallel to the direction of the fibers, are absolutely loaded with gap junctions.
Ah, for communication.
Exactly. Gap junctions form channels directly connecting the cytoplasm of adjacent cells. They allow ions, and therefore electrical current, to pass directly from one cell to the next with almost no resistance.
So the heart muscle cells are electrically coupled.
Perfectly coupled. They function as an electrical syncytium, meaning when one cell depolarizes, the signal spreads rapidly through the gap junctions to all its neighbors. This ensures that large areas of the heart contract almost simultaneously, which is essential for an efficient pump.
So intercalated discs provide both mechanical strength and electrical coupling. Very clever.
Absolutely essential for heart function.
What about the contraction mechanism inside? Same sliding filaments.
Fundamentally, yes. Sarcomeres, actin, myosin, tropomyosin regulation. Calcium binding to TNC is still the trigger.
But the energy demands must be different. The heart never rests.
Never. And it's almost purely aerobic. Cardiac muscle cells are packed, absolutely packed, with mitochondria. They can make up 40% or even more of the cell volume.
Wow, 40%. That's way more than even type I skeletal fibers.
Reflects its utter dependence on constant aerobic ATP production. It cannot tolerate lack of oxygen for long at all.
What fuel does it prefer? Glucose.
It can use glucose, but its primary fuel source is actually fatty acids. It stores quite a bit of fat internally as triglyceride droplets to feed those mitochondria constantly. Also uses lactate readily.
Okay, what about the internal signaling, the SR and T tubules? Is it the same triad system as skeletal muscle?
Similar, but with some key differences. The SR is generally less extensive and less organized than in skeletal muscle. The T-tubules are present, but they are typically larger in diameter.
And where do they invaginate? Still at the AI junction.
Not usually. In mammalian ventricular muscle, the T-tubules tend to invaginate at the level of the Z discs.
Ah, different location. Does this change the SR coupling?
It does. Instead of a T-tubule being sandwiched between two large terminal cisternae, a triad, you typically find one T-tubule associated with just one smaller SR cisterna.
Yeah.
This configuration is called a dyad.
One T-tubule, one SR element, a dyad. Still works for excitation-contraction coupling.
Yes, it still ensures rapid signal transmission for calcium release. But cardiac muscle is also somewhat more reliant on calcium entering the cell from the outside through channels in the T-tubule membrane to trigger the larger release from the SR, calcium-induced calcium release.
Interesting nuance. Now, the heartbeat itself, it's intrinsic, right? Doesn't need nerves to start it.
Correct. The basic rhythm is intrinsic and spontaneous. It's initiated by specialized pacemaker cells, modified cardiomyocytes, in nodes like the SA node. They depolarize automatically.
So what do the nerves do? Sympathetic, parasympathetic?
They modulate the intrinsic rate and force. The sympathetic nervous system speeds up the heart rate and increases contractility. The parasympathetic system, via the vagus nerve, slows it down, but they don't initiate the beat.
One last surprising thing about cardiac muscle, it has an endocrine function. It makes hormones.
It does, specifically the cells in the atria. They contain small secretory granules, usually near the nuclei.
What's inside?
A peptide hormone called atrial natriuretic factor, or ANF, also sometimes called ANP, atrial natriuretic peptide.
What does ANF do?
It's released in response to atrial stretching, usually due to high blood volume or pressure. It acts primarily on the kidneys, promoting the excretion of sodium, natriuresis, and water.
So it lowers blood volume and pressure.
Exactly. It counteracts the effects of hormones like aldosterone and ADH that tend to retain salt and water. It's part of the heart's role in regulating overall fluid balance and blood pressure, quite sophisticated for a muscle cell.
Okay, on to our third and final type, smooth muscle. Found lining organs, vessels, doing the slow, involuntary work.
That's it. Specialized for slow, steady, often sustained contractions. Very different visually from striated muscle.
How do the cells look?
They're typically much smaller, elongated, but they taper at both ends, the classic fusiform or spindle shape. Length varies hugely depending on location, from maybe 20 micrometers in small arterioles up to 500 micrometers, half a millimeter, in the pregnant uterus.
And the key visual feature is?
No striations. The contractile proteins are arranged differently, so you don't see that banding pattern.
Nucleus.
Single nucleus, like cardiac muscle, and it's central, usually elongated, located in the widest part of the spindle-shaped cell.
How are they packed together?
Very efficiently. They form sheets or layers, often with the narrow, tapered end of one cell nestled against the thicker central part of its neighbors. Lots of gap junctions linking them, too, for coordinated action.
You mentioned something about the nucleus shape changing during contraction.
Yes, because the contraction pulls the cell ends inwards quite dramatically, the nucleus often gets squished and twisted, taking on a characteristic corkscrew or pleated appearance. Seeing that is a good sign you're looking at contracted smooth muscle.
Okay, if there are no striations, how is the contractile apparatus arranged? Still actin and myosin?
Still actin and myosin, yes. But instead of organized sarcomeres, the thick and thin filaments form bundles that crisscross the sarcoplasm more obliquely in a sort of lattice-like network. The myosin arrangement is less regular, too.
If there are no Z discs, what anchors the thin filaments?
Structures called dense bodies. These are electron-dense plaques found scattered within the cytoplasm and also attached to the inner surface of the sarcolemma.
What's in them?
They contain I-actinin, the same protein found in Z discs. So functionally, dense bodies are the smooth muscle equivalent of Z discs, serving as anchor points for the thin actin filaments.
Is anything else attached to these dense bodies?
Yes, Importantly, there's also a network of intermediate filaments, typically made of the protein desmin. These filaments also attach to the dense bodies, forming a strong, flexible cytoskeleton that supports the cell and helps transmit the contractile force throughout this oblique network.
So the force generated by actin-myosin sliding is transmitted through the thin filaments to the dense bodies, and then
throughout the cell via the intermediate filament network. The dense bodies attached to the cell membrane also link to extracellular structures and, via associated proteins like adherins, transmit force to adjacent cells. This ensures the whole tissue sheet contracts effectively.
Now, excitation-contraction coupling, how does the signal work here? No T-tubules, you said.
Correct. The cells are small enough and well connected by gap junctions that deep invaginations aren't really needed for rapid signal spread. The SR is also present, but much less organized, more rudimentary.
So how does calcium get released or enter the cell?
Smooth muscle relies more heavily on calcium entering from outside the cell. The sarcolemma has numerous small invaginations called caveolae. Think of them as tiny little pits or pockets.
Like miniature T-tubules.
Sort of functionally analogous, yes. They contain ion channels, including calcium channels, and are thought to play a role in sensing stimuli and organizing the signaling molecules needed to control calcium release from the nearby sparse SR and calcium entry from outside.
Okay, calcium levels rise in the sarcoplasm. What happens next? Because you said there's no troponin C.
Exactly. This is the biggest difference in regulation. Troponin is completely absent in smooth muscle. The regulation is myosin-based, not actin-based.
How does that work?
It involves a protein called calmodulin. When intracellular calcium levels rise, calcium binds to calmodulin.
Like it binds to troponin C in striated muscle.
Similar initial step, yes. The calcium calmodulin complex then activates an enzyme called myosin light chain kinase, or MLCK.
Kinase, so it adds a phosphate group.
Precisely. The activated MLCK phosphorylates one of the light chains located on the myosin head.
And the phosphorylation is the trigger.
That's the key. Phosphorylation of the myosin light chain is required for the myosin head to interact with actin and start the cross-bridge cycling in smooth muscle. Dephosphorylation by another enzyme, myosin light chain phosphatase, turns it off.
So it's a chemical switch, phosphorylation, rather than a physical blocking, unblocking mechanism, tropomyosin.
Exactly. This multi-step biochemical cascade is inherently slower than the direct conformational change triggered by calcium binding to troponin.
Which explains why smooth muscle contraction is generally slower to start and stop.
Yes, but it's also very efficient. It allows for sustained contractions with relatively low ATP consumption, which is perfect for things like maintaining blood vessel tone or slow peristalsis.
How is smooth muscle controlled? Nerves, hormones.
All of the above, making it very versatile. It's controlled by the autonomic nervous system, both sympathetic and parasympathetic, by circulating hormones like oxytocin on the uterus or epinephrine on blood vessels, and also by local paracrine signals, like substances released in the gut wall.
Does it have NMJs like skeletal muscle?
Not the same well-defined one-to-one junctions. Instead, autonomic nerve axons passing through smooth muscle tissue have periodic swellings along their length called varicosities.
Little bumps on the nerve fiber.
Yeah, these varicosities contain synaptic vesicles with neurotransmitters, like acetylcholine or norepinephrine. When the nerve fires, transmitter is released from these varicosities and diffuses over a wider area to reach receptors on multiple nearby smooth muscle cells.
A less precise, more diffuse innervation.
Right, and the signal then spreads efficiently through the tissue via all the gap junctions, leading to coordinated contraction of sheets or bundles of cells. Perfect for things like peristalsis.
One last point, smooth muscle can make matrix components, like fibroblasts.
Yes, another aspect of their versatility. Unlike highly specialized striated muscle cells, smooth muscle cells retain the ability to synthesize and secrete components of the extracellular matrix, including collagen, elastin, and proteoglycans.
So they help maintain the structure of the tissue they're in.
Exactly. They play a significant role in building and maintaining the ECM, especially in the walls of blood vessels and organs. They're not just contractile cells. They have this important synthetic role, too.
Okay, we've covered the motors, but the brain needs feedback, right? How does the CNS know what the muscles are doing? Proprioception.
Crucial for coordinated movement and posture. The musculoskeletal system is loaded with sensory receptors sending constant updates back to the brain and spinal cord.
What are the main ones within the muscle itself?
Two key players. First, the muscle spindles. These are fascinating little sensory organs scattered within the muscle belly, lying parallel to the main contractile fibers, the extrafusal fibers.
What do they look like?
They're small, maybe a few millimeters long, and encapsulated by a modified connective tissue sheath derived from the paramecium. Inside this capsule are a few specialized thin muscle fibers called intrafusal fibers.
Intra means within the spindle, extra means outside.
Exactly. Sensory nerve endings wrap around these intrafusal fibers. Their job is to detect changes in the length or stretch of the main muscle.
So when the muscle stretches, the spindle signals.
Yes. Stretching the main muscle also stretches the intrafusal fibers, activating the sensory nerves. This information goes to the CNS and is used in stretch reflexes, maintaining muscle tone, and providing conscious awareness of limb position.
Okay, so spindles detect length. What's the second sensor?
The Golgi tendon organs, or GTOs. These are located at the other end, right at the myotendinous junction, where the muscle transitions into the tendon.
And what do they detect?
They detect changes in tension or force generated by the muscle contraction. They're situated in series with the muscle fibers.
How do they work?
GTOs are small encapsulated structures containing collagen fibers from the tendon and sensory nerve endings intertwined with them. When the muscle contracts and pulls on the tendon, it squeezes these nerve endings.
Triggering a signal about force.
Exactly. High tension activates the GTO. This signal typically acts inhibitorily on the motor neurons supplying the muscle. It's primarily a protective reflex, preventing excessive force generation that might damage the muscle or tendon.
So spindles monitor length, GTOs monitor tension. Constant feedback loop.
A very sophisticated sensory feedback system.
Finally, let's wrap up with regeneration. How well do these different muscle types repair themselves after injury? Let's recap. Skeletal muscle.
Limited regeneration. The mature, multinucleated fibers can't divide. Repair depends entirely on those satellite cells activating, proliferating, and fusing. Good for minor damage, but significant injury leads mostly to scar tissue.
Cardiac muscle. The heart.
Very poor regeneration. Essentially none in adult mammals. Cardiomyocytes don't divide, and there are no satellite cells. Damage, like from a heart attack, myocardial infarction, is permanently replaced by non-contractile fibrous scar tissue formed by fibroblasts.
That's why heart attacks cause lasting damage.
Precisely. The lost muscle isn't replaced.
And smooth muscle.
Good regeneration. This is the winner in the repair department. The simpler mononucleated smooth muscle cells can and do undergo mitosis to replace damaged cells.
So they can actually divide and repopulate an injured area.
Yes. Plus, other cells like parasites, cells associated with capillaries, can sometimes differentiate into smooth muscle cells, particularly in blood vessel repair. So smooth muscle has a much more robust regenerative capacity compared to the striated types.
Hashtag tag outro.
Wow, that was definitely a deep dive. We've gone from the whole muscle organization down to the molecular dance of actin and myosin. Covered the lingo, the structures, the controls, intricate stuff. But hopefully, breaking it down piece by piece makes it clear. The key really is linking the structure you see under the microscope to the function it performs.
Absolutely. And appreciating those key differences. For anyone aiming for that elite understanding, remembering the hallmarks is crucial. Skeletal, voluntary peripheral nuclei, striations, the triad, troponin C control.
Cardiac, involuntary, central nucleus striations, those vital intercalated discs with their desmosomes and gap junctions, the diet system, massive mitochondrial content.
And smooth. Involuntary, fusiform shape, no striations, single central nucleus, dense bodies anchoring oblique filaments, and that unique calmodulin MLCK activation switch.
Mastering those details, the epimysium channeling force, the role of giant titin, the specifics of TNC versus MLCK regulation, that's what elevates understanding beyond the basics. That's the professional level grasp.
And speaking of deep understanding, that point about cardiac regeneration or the lack thereof in humans compared to other animals, that's really striking.
It is. The sources mention how newborn mice and even adult fish and amphibians can regenerate heart muscle after injury, often without satellite cells. Yet, adult mammals, including us, can't. We just scar.
So if the basic building blocks, the sarcomere machinery, are essentially the same across these species, why can they regenerate heart muscle and we can't?
That's the million-dollar question, isn't it? Is the limitation in adult human hearts purely because we lack the right stem cells or precursor cells? Or is there something more fundamental?
Like a developmental program that gets switched off, or an environment in the adult heart that actively suppresses regeneration, even if the potential exists.
Exactly. Is it a missing go signal or an active stop signal? Unlocking that could revolutionize treatment for heart disease. It suggests there might be an ancient intrinsic regenerative ability that our adult hearts have lost access to.
A really provocative thought to end on. Figuring that out would be a monumental leap.
Well, thank you for navigating us through that incredible complexity.
My pleasure. It's a fascinating tissue system. And thank you all for joining us on the deep dive. Hopefully, you feel you've mastered muscle tissue just a little bit more today. We'll catch you next time.